Hello everyone. In this podcast we're going to talk about some basic introductory terms with genetics as are outlined on your note-taking guide. I'm going to begin by talking about what we call the central dogma. This is a concept that we will come back to frequently throughout the semester, but I think it's good for us to kind of introduce it right now. In its simplest form, it is just taking DNA, having it transcribed, into messenger RNA, then having the messenger RNA translated into protein. And the protein, for all practical purposes, is that particular organism's trait. In some cases, the messenger RNA will be a trait as well. Which brings me to another important point to make here, and that is that the central dogma is a general guideline. We will learn throughout the semester that it's not an absolute dogma, that sometimes we'll find that messenger RNAs can behave like proteins. That is, they can have a function that we might have thought only for proteins. We can also learn that sometimes the RNA is the genetic material. So this is a general guideline, and it holds up in most cases. Let's now talk about a specific example related to the central dogma here. So let's draw a piece of DNA. A very simplistic model here. And let's say a chunk of this DNA is a gene. So let's write gene here. But specifically, let's talk about a particular gene. And let's talk about this gene called GLUT, G-L-U-T 2. GLUT2. Now this gene, GLUT2, by itself is just a blueprint. And it doesn't have a particular function all by itself. Its function is just to exist and to carry the message necessary to make a particular protein. So this gene GLUT2 will be transcribed, and it will make a messenger RNA. And this will be our symbol for messenger RNA. And it's messenger RNA that's specific to GLUT2. GLUT2, messenger RNA, then now will be translated. And it will be translated into a very specific protein that is unique to just this one protein, GLUT2. And I'm just going to draw it as this big globular structure here. But this would be GLUT2 protein. And this protein has a very specific function. I'm going to draw it here. Let's say this is our cell here. GLUT2 is a receptor on the outside of the cell that brings in, we could also call it a transporter. It brings glucose into the cell, which is really important for that cell's metabolism. Now I want to bring up another term here, and that term is genetic variation. Let's draw another piece of DNA here. And on this piece of DNA, I'm going to put a little asterisk by it to symbolize that this piece of DNA, which is also the GLUT2 gene, but it has a mutation on it. So I'm going to put a minus sign there. And so this is part of what variation is. That there are differences in inherited traits found within individuals of a population. Let's go ahead and write that down. So genetic variation is equal to the differences in inherited traits found in individuals in a population. So within this make-believe population here, we have some individuals who have the GLUT2 gene that doesn't have a mutation, and it makes this protein that's found in the cells that's important in glucose metabolism. Well, this other individual in this population has a mutation in GLUT2. GLUT2 mutant will still be transcribed and will still make a messenger RNA. And don't worry too much about some of these terms. We'll come back to a lot of these terms: transcription, translation, messenger RNA, protein, all that much later. This is just kind of introduce this concept of genetic variation. So we'll still make this protein, this messenger RNA, and it has a mutation in it. So we'll put a minus sign there. And it's still going to make the protein, except now the protein is going to be just a little different, in such a way that I'm just drawing it a little different to illustrate that now when glucose tries to enter the cell, it still can get in, but not as efficiently, as I'll show with the dotted line here. So it can still get in there, but not as efficiently. So now we have a genetic variation where we have non-mutated and a mutated cell producing a protein that's a little bit different. It generates that variation within the population. I'm going to erase this definition for genetic variation at the bottom because there's another really important part here I want to talk about. Now, one thing I didn't mention was that this GLUT2 gene, when it's mutated, is linked to diabetes type 2. Now, if you have this mutation, it doesn't mean you're going to get diabetes type 2. It means that you have the gene that causes you to have an increased risk of developing diabetes type 2. So that brings us to the important point of what controls a trait. We've hinted to it, but we haven't actually said it. So a trait is caused by a combination of genetics plus the environment. In this example here, the genetics gave this individual here on the bottom a mutation that, under certain environmental conditions, can lead to diabetes type 2. So we have the genetic variation, and under certain environmental conditions, that can lead to the disease of diabetes type 2. This individual, if they monitor how many carbohydrates they were eating and if they exercise properly, it's quite possible that they would never develop diabetes type 2. Or they would at least delay the onset of diabetes type 2. In this example, I focused on a human condition and a human disease. It's important to remember that these terms of traits and genetic variation and everything else we talked about is found throughout all organisms, including bacteria, archaea, eukaryotes, and within eukaryotes, all the animals and all the plants and the fungi. Which brings me to our next point, and that is people who are interested in complex processes, such as diabetes type 2, and how that affects humans, don't often study these diseases or other complex processes in humans or other large animals. Instead, scientists and geneticists will focus on genetic model organisms. So let's talk about that for a moment. So up here, let's write genetic model organisms. And as I said before I erased the board, that we use these genetic model organisms to study complex processes using a much simpler organism. This helps us break down these larger processes into simpler questions. So model organisms typically have certain traits. These include, often anyhow, they're small. It's not required, but it certainly is very helpful if that model organism's genome is sequenced. Typically, they will have a short generation time. For instance, if you wanted to study a scientific question and an organism had a generation time of 3 hours as opposed to an organism that had a generation time of every 50 years, well, the one that has a generation time of 3 hours is going to be more useful. You're going to get more information from that organism. These organisms can have their genomes somewhat easily manipulated. Because of their size and their simplicity, often model organisms are cheaper to work with. And this isn't necessarily a trait, it's just a simple fact that since all these organisms are genetically related, what we learn in a simpler system, we can apply to a more complex system. So I'm just going to write here genes evolutionarily conserved. Every now and then you will get new genes popping up, but even the new genes have some sort of evolutionary conservation with the pre-existing genes. Before we move on, I just want to say one more thing, and that is just provide some examples of genetic model organisms that have all of these characteristics. So E. coli, a very common bacteria, Saccharomyces cerevisiae, which is your baker's yeast, the same yeast that you use to make bread, beer, wine. Drosophila, which is a fruit fly. C. elegans. And the last one here I want to mention is Arabidopsis. C. elegans, I should have mentioned, is this microscopic worm that lives in soil, and Arabidopsis is a plant. Throughout this semester in lab and in lecture, we'll talk about all of these in lecture, but in lab you'll have an opportunity to work with E. coli, Saccharomyces cerevisiae, Drosophila, and C. elegans. Unfortunately, we won't be able to work with Arabidopsis in the summer session. We can lump geneticists into one of three categories, though it's probably not a good idea, but let's go ahead and do it just for a moment here. Transmission geneticists, sometimes we call this Mendelian genetics. Questions a transmission geneticist might ask is, how are genes transmitted from parent to offspring? For instance, if you had a red-eyed fly and you cross that with another red-eyed fly and you ended up getting a fly without red eyes but rather white eyes, a transmission geneticist would investigate why this is possible. Why are not the progeny red-eyed? Next we have molecular genetics. I guess I should have wrote genetics under here. Essentially, they want to know is, how does the genetic material work at the molecular level? So a molecular geneticist is going to be interested in a question like, how does this gene that we talked about on the last couple slides ago, how does this gene that gives you the protein GLUT2, how does this happen? What are the molecular mechanisms that allow a piece of DNA to carry a message to make a protein. They would also be interested in how the environment affects how this protein is made and what it does with that protein. A population geneticist they're interested in the genetic variation in a population. And how the frequency of genes change with evolution. So a population geneticist is going to be interested in looking at that population and determining the different kinds of GLUT2 genes there are and how the frequency of those genes change over time. Over time, one kind of gene or another may have a particular advantage to that population. And whatever that advantage may be, or disadvantage may be, that's the kind of question the population geneticist is interested in. Though we have listed all three of these types of geneticists, it's important to remember that it would not be reasonable to think that all transmission geneticists only study questions of transmission and all molecular geneticists only study questions of molecular genetics, and same thing with population geneticists. The fact of the matter is that the three different fields of genetics merge with each other, and the lines that separate them are quite blurry. It's quite common that somebody's interested in transmission genetics will also be interested in questions about that involve the molecular level. And the same is true with the other two examples. So that ends this podcast. And just as a quick summary here, what you should have gotten from this podcast is what the central dogma was and how to explain it. You should understand genetic variation and how that genetic variation is important in having a large diversity of characteristics within a population. You should have gotten that traits are caused by genes plus the environment. You should have learned what makes a model organism a good model organism, the traits of it, and some examples. And lastly, the different kinds of geneticists. Okay, well that ends today's podcast. If you have any questions, please let me know. See you in class. Bye.